Welcome back to The Explainer. Today, we're cracking open a literal medical cold case, one that's been baffling doctors and researchers for decades. We're zeroing in on a highly fatal malignancy that has somehow managed to slip right past modern medicine's absolute best defenses. For years, oncologists have thrown standard therapies at it, but the outcomes? Stubbornly grim. But here's the exciting part. Recent breakthroughs in cell culture research and medical imaging are finally shedding light on what this disease actually is. So let's look at the clues, follow the data, and solve this mystery together, shall we?
It all kicks off with one big, glaring question. Why on earth are we failing to cure so many cases of invasive lobular breast cancer, or, ILC for short? Think about it. We've seen incredible advances over the last few decades, right? Precision medicine, targeted chemo, advanced radiation, yet a massive number of patients with classic ILC are still slipping through the cracks. It's a question that practically begs for an answer. And as we're going to see, the problem might not actually be our tools. It might be our fundamental understanding of what we're even fighting in the first place.
Okay, let's dive into this and look at the hard data. What you're seeing here is basically the mathematical proof of our cold case. Along the bottom, you've got the time since diagnosis, stretching all the way out to nearly 20 years. And on the side, we have cumulative survival. Now really look at these two lines. The blue one tracks diffuse ILC cases from 1996 to 2010. The red one? That's the very next decade, 2011 to 2022. Notice anything? They are virtually identical. I mean, despite the massive leaps in medical tech between those two eras, the long-term survival for women with classic ILC has stayed dismally stagnant, hovering right around 60%. The therapies we've been relying on are just flat out failing to control this. To understand why, we've got to take a quick trip to the past. So, for over 75 years, literally 3/4 of a century, the medical community was operating on a pretty massive assumption, one made way back in 1941 by researchers Foote and Stewart. They theorized that this cancer started in the epithelial cells of the breast lobule. But here is the crazy catch. Even they admitted they didn't have the anatomical proof. It was just a theory. But somehow, it became accepted fact. Fast forward to today, and brand new cell culture evidence has completely blown that assumption out of the water. This cancer doesn't start in the epithelial cells at all. Actually, it originates from mesenchymal hybrid stem cells. Basically, for decades, we've been going after the completely wrong suspect.
Because of this massive breakthrough, researchers are now pushing to reclassify classic ILC. The new proposed term is breast cancer of mesenchymal origin, or BCMO. Now, you might be wondering, why does a name change matter so much? Well, think of it like this. Trying to treat a mesenchymal stem cell cancer with therapies designed for epithelial cancer, it's like trying to fix a busted pipe by rewiring your living room. You're messing with the completely wrong system. Mesenchymal stem cells are notorious for lacking E-coherent, which is basically cellular glue. Without that glue, these cells can literally shapeshift and just slip right past our standard epithelial treatments. No wonder those survival curves haven't budged.
These microscopic clues reveal exactly how stealthy this disease actually is. Take a look at the mammogram on the left side of the screen. There's no neat, easily recognizable lump, right? No obvious calcifications. Then, moving to the middle and right, we get a closer look at the tissue samples and specimen radiograph. What you're seeing here is something called non-calcified architectural distortion. Because of its mesenchymal origin, this highly fatal extralobular form of BCMO doesn't form a solid mass. Instead, it spreads out like a web. It is incredibly sneaky. It just pulls and distorts the tissue architecture, making it practically invisible to standard screening that's actively looking for dense, solid masses.
So the absolutely crucial takeaway here is that extralobular BCMO is the ultimate stealth suspect. It starts out in the extralobular connective tissue and just spreads freely. And because it doesn't form that easily detectable lump we just talked about, surgical removal is incredibly tricky. A lot of times it's incomplete because surgeons quite literally cannot see the full extent of the disease on standard imaging. But here is the scariest part. The standard molecular biomarkers the tests doctors usually rely on to predict how a cancer is going to behave, they often come back looking deceptively favorable. A patient's chart might scream optimistic outcome, while in reality, this stealthy, web-like disease is telling a much, much darker story. A real tale of two suspects. Yep, it turns out we aren't just hunting down one culprit. We're actually dealing with two totally distinct forms of classic ILC that up until right now have been confusingly lumped together under the exact same name. Let's introduce suspect number 2.
intralobular BCMO. Now, what's really interesting about this slide is the visual contrast. It's basically the smoking gun. Look over at the left, boom, right away you can see a solitary, spiculated mass. It is very localized. But the undeniable proof is in the high-power microscopic view on the right. See that dark brown staining the solid black arrows are pointing to? Those are your normal, non-malignant epithelial cells inside the breast acini. They test positive for E-cadherin, that cellular glue. But look right next to them. See the cells marked by the blue arrows? Those are the BCMO cancer cells. They are negative for E-cadherin. They look completely different because they are fundamentally different. They originate from the mesenchyme and are literally surrounding the healthy epithelial cells.
Let's quickly summarize how this intralobular suspect operates. Unlike its extralobular cousin that spreads like wildfire through connective tissue, this form starts in the intralobular mesenchyme and actually forms small, distinct colonies. Because it clumps together around normal breast structures, it shows up as spiculated or circular masses on imaging. Now, this is a real game changer. It means doctors have a massive advantage. This form is way easier to spot early on a standard mammogram, which totally changes the playing field for the patient.
So, how do all these microscopic differences actually translate to real-world patient outcomes? Listen, this is not just some academic debate about cell shapes. It is literally a matter of life and death. Look at this massive mathematical difference between our two suspects over a 19-year period. That top blue line, that tracks the patients with the localized, easier-to-detect intralobular BCMO. They're sitting at an 84% survival rate. Fantastic. But the orange line below it, that tracks the stealthy, freely spreading extralobular BCMO. Survival plummets down to just 56%. Lumping these two wildly different diseases into one catch-all bucket of classic ILC, it totally masks reality and, frankly, misguides treatment.
Which brings us to a pretty powerful verdict, straight from the researchers investigating this exact cold case. They hit the nail on the head, stating, "The accepted terminology and assumptions of the origin of diffuse and extensive classic ILC must be reconsidered if we are to improve the poor survival rate of this misunderstood malignancy. We absolutely cannot afford to just coast on a 75-year-old assumption anymore. Recognizing the true mesenchymal stem cell origin of this cancer, it's not just a matter of semantics. It is an urgent, massive call to completely overhaul how we diagnose, research, and ultimately treat this disease." Which leaves us with one final, pretty provocative thought. If we now have the visual and cellular proof to finally solve this 75-year-old medical mystery, how quickly can the medical community pivot its treatments to stop this stealthy killer? The evidence is right in front of us, the biology makes sense, and the data is screaming for action. It really challenges all of us to continually question the things we just assume to be true. Because, as you can see, breaking those old assumptions is almost always the first step toward saving lives. Thanks for tuning into this explainer, and remember to always keep questioning the evidence.